Thank you first for your compliment. Thank you, Professor Lung, for inviting me to this symposium and giving me the opportunity to present my studies in here. A large fraction of my career was actually devoted to understanding of oocyte maturation. Completion of the meiotic cell cycle and preparation of the oocyte for fertilization has indeed a major role in successful reproduction. Nevertheless, I would like today to introduce you to another play in this fascinating show. And this player is Vasorin. I will start my talk by introducing you to Vasorin, and this will be followed by description of its mechanism of action. And since Vasorin action is mediated by TGF-beta, I will dwell shortly on the TGF-beta superfamily members. This will be followed by presentation of my research aim, experimental model, results, and I will, of course, wrap up with the conclusions. Vasorin is a type 1 transmembrane glycoprotein, which shows a very high level of identity between murine and human, both the messenger RNA and the amino acid composition of the protein, which means that it is a highly conserved molecule. It was first identified in vascular smooth muscle cells in 2004. It took 7 years to describe or to realize the mechanism of action of Vasorin and show that it actually regulates the TGF-beta signaling pathway by a way by which it, if this is the Vasorin molecule, as I mentioned, it is a transmembrane molecule. It is being cleaved by a metalloproteinase, ADAM17, and then the soluble Vasorin binds the TGF-beta, acting, competing with on the TGF-beta receptor for binding of TGF-beta, thus lowering the availability of TGF-beta. In fact, Vasorin acts as a negative regulator of TGF-beta. As I mentioned, TGF-beta is a superfamily, and some member of which are activin, which is involved in primordial follicle assembly, GDF9 and BMP15 that are required for early stages of folliculogenesis, AMH that we all know, which inhibits primordial follicle growth, and TGF-beta1 itself, which is involved in ovulation, corpus luteum function, and angiogenesis. As TGF-beta family member has so many roles in different aspects of folliculogenesis, we were highly intrigued to look for Vasorin, which is an upstream regulator of TGF-beta. And I don't have to tell this audience, this life history of the ovarian follicle is quite familiar to this audience. I will just mention that it starts around birth with a huge, I don't know the number, but a huge pool of primordial follicle, and the follicles are recruited throughout the lifespan of the female until sexual maturation. Some of them are going up to the antral follicle, and most of them die by atresia. And after sexual maturation, there is cyclic recruitment of follicles from this pool to go all the way to ovulation by selection and dominance. At this stage, also many follicles undergo atresia. And the big question that has been raised several times this morning in the many presentations up to now relates to the mechanism that controls the number of follicles recruited for growing and ovulation, thus maintaining the ovarian reserve. And our research aim was to elucidate the role of Vasorin in ovarian physiology and female fertility, focusing in particular on its potential involvement in the protection of the ovarian reserve. And in addition to wild-type mice, we used another 2 mouse models. One was the Vasorin systemic knockout mice, and the other one was Vasorin conditional knockout mice, in which the deletion was directed at the ovarian granulosa cells. I will provide more details about this model later on. The important point is to remember that the deletion here is only target or limited to the ovarian granulosa cells. And our experimental design, the first experiment that we designed was to look if the ovary expresses Vasorin at all, and then the second question at that stage was to see whether Vasorin expression in the ovary is regulated by LH. And as you see in this slide, both by using in situ hybridization and immunohistochemistry, we show that messenger RNA and protein of Vasorin are expressed in the ovary. And we also demonstrated that Vasorin expression in the ovary is upregulated by LH, you see here. And in this close-up picture, you see that expression of the messenger RNA of Vasorin, at least, is limited to the granulosa and cumulus cells. Neither the oocyte nor the theca cells express Vasorin. And using RT-PCR, we were able to profile the kinetics of Vasorin expression in response to LH. And you see here in the samples of whole ovaries, as well as in isolated granulosa cells, that the expression of Vasorin peaks at 4 hours after LH. A similar pattern of expression was also obtained or observed looking at the protein level, and you see here that 4 hours after HCG, there is upregulation of Vasorin protein expression. And the downstream elements that are involved in the LH signaling are EGF receptor, PKA, MAP kinase, and protein and progesterone receptor. We used specific pharmacological inhibitors to find out which of these components is involved in LH-induced Vasorin expression. And as you see here, it is the EGF receptor and the MAP kinase that mediate the effect on LH on upregulation of Vasorin expression in the ovary. And the second aim of the second experiment in our study was to find out whether Vasorin has any role in ovulation, and this was based on the fact that LH is upregulating Vasorin expression. And again, we used our 2 animal models, the conditional and the systemic knockout mice. In the conditional knockout mice, we see that Vasorin expression or depletion of Vasorin from the ovary does not impair their fertility. There are the number of pups per liter as well as the average accumulated number of pups are similar in the conditional knockout and wild-type mice. You see here, this was accumulated throughout 6 months. 6 months. And we were very surprised to find out that Vasorin regulates the ovulation size in a manner by which it increases or almost doubles the number of ovulated oocytes in the mouse. And one way to, this experiment was done on the conditional mouse model, and since the systemic knockout mice do not live long enough to reach sexual maturation, that they die at about 3 weeks due to severe kidney failure, we did the following experiment. We recovered the ovaries of the systemic knockout mice, and we transplanted them under the kidney capsule of wild-type mice. We let them to recover for a week, and then the mice were subjected to the common protocol of induction of ovulation, PMSG followed by HCG, and as you see here, and the only way to look for ovulation at this model is to count the number of corpora lutea, and as you see here, this protocol reproduce our finding using the conditional knockout mice, and we have higher number of corpora lutea in the absence of Vasorin. And trying to explain this finding, we stain the ovaries by tunnel for apoptosis, and we see that there are less apoptotic antral follicles in the conditional knockout mice, and this was quantified here. And we concluded that it is possible that a larger pool of antral follicles may account for the higher size of ovulation in the conditional knockout mice. And the canonical downstream target for TGF-beta is SMAD2 and 2,3. So we assume that in absence of Vasorin, the level of activated or phosphorylated SMAD2 will be upregulated. And indeed, as you see here by the fluorescent antibody for phosphorylated SMAD, you see that indeed in absence of Vasorin, there is an increase in the level of activation of SMAD, and this was quantified here. The following experiment that was conducted was the analysis of the role of Vasorin in early folliculogenesis. And for this purpose, we counted the number of follicles in sections of ovaries recovered from either conditional or wild-type animals. And as I already mentioned, there is a higher number of Vasorin, of antral and secondary antral follicles in the Vasorin knockout mice. In the absence of Vasorin, a similar number of the secondary follicle, and quite surprisingly, we see a much lower number of primary and primordial follicle in the absence of Vasorin in the ovaries of 15-day-old conditional knockout mice. And to explain this finding, let me go for the details of our conditional knockout model. And the deletion of Vasorin in our model is coupled to the expression of CYP19, which takes place only in growing follicles. So follicles that are smaller than growing follicle, I mean primordial and primary follicle, do not express CYP19. Therefore, they do contain the Vasorin even in our conditional knockout system. Thus, the lower number of small follicles is due to the lack of Vasorin in growing follicle. And what does it mean? We try to provide a working model for Vasorin in this cartoon. Vasorin is in the wild-type animal. I'm not talking about now about the conditional knockout. The wild-type animal, the going and the early antral follicle do express vasorin. This vasorin is suppressing either the recruitment of the primary follicles into the growing pool and or preventing atresia in these follicles. And to summarize, vasorin, I hope that I convince you today that vasorin is expressed in the ovary. Vasorin expression is regulated by LH. The effect of LH is mediated by MAP kinase and the EGF receptor. And vasorin controls the size of ovulation. Vasorin regulates the TGF-beta signaling pathway in the ovary, and vasorin maintains the ovarian reserve in early folliculogenesis. And our conclusions so far are that vasorin is a new central player in folliculogenesis, and as vasorin is evolutionary conserved, and then it has been recently identified in human follicular fluid by there's one publication that shows this, and recently we also were successful in determining vasorin in human follicular fluid. So it may be a target for possible clinical implication such as fertility preservation. This study was part of the PhD thesis of my former student, Nitzan Rimon. It is now being studied by a present PhD student, Daphna Keter. It was published this year in FASEP Journal, and it was recommended by the 1000th faculty as being a special significance in its field, and I thank you for your attention.